Hello everyone, I welcome you all to this 18th lecture of this course. So this 18th lecture is on cellular uptake mechanism of nanomaterials. So in this lecture we are going to learn what are the various multiple portals available for cellular entry, and also how the nanoparticle can enter the cells through phagocytosis and phagocytosis process. And here we are also going to learn how the nanoparticle's properties influencing the cellular uptake. So the design of smart multifunctional nanosystems for intracellular imaging and targeted therapeutic applications require a thorough understanding of the mechanism of nanoparticles. Okay, so because the nanoparticles is considered as one of the important delivery vehicle for delivering the drugs and genes, because the site specific delivery of drugs and therapeutics can significantly reduce the drug toxicity and increase the therapeutic effects. So here, so when we design any nanomaterials, it is better to understand the complete mechanism, how it is taken up by the cells, because when you understand the complete mechanism, then we can easily target to the particular cell, and also we can release the drug to increase the therapeutic efficiency. And for biological and clinical application, so the ability to control and manipulate the accumulation of nanoparticles for an extended period of time inside the cell can lead to improvements in diagnostic sensitivity and therapeutic efficiency. For example, if you are making a nanomaterial for diagnostic applications, so it should enter the particular cancer cell and it has to stay for more time, so that we can get more signal and we can detect the cancer in the early stage. And also this illustrating the exocytosis mechanism and metabolism of nanoparticles in the cells could lead to a better understanding of nanoparticle toxicity. That is, if the nanoparticles are trapped in the vesicles and leave the cells intact, they are unlikely to induce cellular toxicity. For example, if a nanoparticle is entering into the cell, okay, and it is not entering the nucleus or the cytoplasm and it is leaving the cells intact, then it will not induce a cellular cytotoxicity to kill the particular cells. Okay. So it is better to understand the mechanism, how it can enter into the cell and what is the subsequent process, how it can induce a therapeutic efficiency. Those things has to be understood thoroughly before we take the nanomedicine to the clinical application. So let us see how these nanoparticles enter in the body. So here this nanoparticle enters in the body through the dermal, ocular and respiratory as well as the gastrointestinal systems. So once it's entered the biological environment, so there will be formation of protein corona. Let us see what is protein corona. So in the biological environment, the nanoparticle will adopt a biological identity with the formation of protein corona on its surface due to adsorption of layers of proteins and small molecules such as amino acids and sugars. And the inner hard corona rapidly forms in seconds and strongly attached to the nanoparticle surface, and the secondary soft corona is found atop, so which can take hours to equilibrate due to its high sensitivity to the external environment composition and conditions. So once the nanoparticle enter into the cell, then what happens is like there will be formation of protein corona. So proteins and amino acids will form a layer on the top of this nanoparticle. The first layer is called as hard corona, which is strongly attached to the nanoparticle, and followed by that there will be a soft corona. Okay. So here you can see here the physical properties of nanoparticles, including the size, shape and surface chemistry, so which is going to affect the final corona composition. Okay. And based on that corona composition, the toxicity effect will be varied. Okay. So you can see here this is your hard corona, which is tightly attached to the nanoparticle surface, and followed by that you are having the soft corona. So the soft corona, it is the outer soft corona that is presented to and directly interact with the cell rather than the original nanoparticle surface. So this can pose a challenge while designing specific targeting nanoparticle. So here, so this soft corona is going to interact with your cell, okay, not the nanoparticle charge or anything. So based on the soft corona, it is going to give the therapeutic effect. So when you design any nanoparticles for a targeted delivery, so you have to consider the effect of this soft corona also. So let us see the multiple portals of cellular entry. So nanoparticles are of similar size to typical cellular components and can efficiently intrude the living cells by exploiting this cellular endocytosis machinery, and it can result in the permanent cell damage. So only specialized cells such as macrophages are capable of phagocytosis, a form of endocytosis in which the cells engulf larger particles, and almost all the cells can internalize nanoparticles by phagocytosis. Okay. So when the nanoparticle goes into the body, there is a cell called macrophages. So that will uptake the nanoparticle by a process called phagocytosis. The phagocytosis process means cell eating. So and most of the other cells will be taken up the nanoparticle by a process called phagocytosis. Phagocytosis means cell drinking. So let us see the mechanism of endocytosis in details. So this endocytosis is divided into phagocytosis and phagocytosis. As I told you earlier, the phagocytosis is cell eating process and the phagocytosis is cell drinking process. So under this phagocytosis, again there are subclasses like macrocytosis and clathrin dependent and caveolus dependent and clathrin and caveolus independent. So this phagocytosis is particle dependent, okay, and this phagocytosis again subdivided into 4 categories like macrophagocytosis. So here the size of the vesicle will be more than 1 micrometer, and this clathrin mediated endocytosis, here the size of the vesicle is 120 nanometer, and this caveolin mediated endocytosis the size is 60 nanometer, and in the case of clathrin and caveolin independent endocytosis the size of the vesicle is 90 nanometer. So let us see this mechanism in detail. So first one is phagocytosis. So here the specific endocytic pathway predominantly occurred in phagocytes such as macrophages and neutrophils and monocytes. Okay. So these are the cells will be involved in this phagocytosis process, and so relatively large particles are more likely to take this way. So the bigger size particles will be taken up by the cells by phagocytosis process, and nanoparticles which adopt this way of entry into cells need to be recognized by the opsonin firstly. So the opsonins are like immunoglobulin or complement component and blood serum proteins. Okay. So when the nanoparticle enter into our body, what happens is this immune system will try to come and attack it. So this immune system has to get activated. For that, on the top of the nanoparticle there will be some opsonins will be attached. Opsonins like antibodies or complement protein or blood serum proteins that will be attached to the nanoparticle. So once it attached, this macrophages will go to the nanoparticle and it will engulf. So you can see here. So this is your nanoparticle. On the top of the nanoparticle, some signals opsonins are attached. Okay. So once it is attached, so this macrophage will go and eat that particular nanoparticles. So let us see the phagocytes in details. So these opsonized nanoparticles bind to the cell surface and interact with the receptor, and it will induce a cup shaped membrane extension formation. Okay. So this membrane extension enclose the nanoparticles and then internalize them, forming the phagosomes, so which have a diameter of 0.5 to 10 micrometer, and finally the phagosomes move to fuse with the lysosomes. Okay. So then the cargo contained in the phagosomes will be destroyed by acidification and enzymolysis in the lysosomes. So therefore to produce the desired effects, nanomedicine should bypass this route to avoid the degradation. So if you are making some nanoparticle for therapeutic applications, so that has to escape from this phagocytosis process, then only it can reach the target cell and it can deliver the drug. So you can see the phagocytosis process in details. See here, this is your nanoparticles or nanoboards, and it is having this opsonin, okay, that is your ligand, and when it binds to this macrophage cells which is having the receptor, so and it form a vesicle that is called as phagosomes, and this phagosomes will fuse with the lysosomes, okay, and inside the lysosome you are having lot of digestive enzymes. So and this will degrade these nanomaterials, then it release the degraded nanomaterials. So if your nanoparticle is carrying a therapeutic molecule like anti-cancer drug or anything, so if it is taken up by these macrophage cells by phagocytosis process, then it will degrade the nanoparticles, then it cannot induce the desired therapeutic effect. So let us see the next mechanism, that is called as pinocytosis. Okay. So it is called as cell drinking. So it is a non-specific route where the membrane invaginates and engulf the materials and then pinches off, producing vesicles of sizes between 0.5 and 5 micrometer. So this mechanism sometimes referred to as macropinocytosis for larger vesicle formation, and it is primarily used for the uptake of fluids and other essential materials required for the cells such as salts, glucose and amino acids. So this is a normal process for uptaking the salts and glucose and amino acids, okay, and if any foreign particle comes, nanoparticles, that can also taken up by this pinocytosis process. And again this pinocytosis process classified into clathrin dependent, caveolus dependent, macropinocytosis, or caveolus and clathrin independent based on the proteins involved in this pathway. So let us see clathrin dependent endocytosis, that is CME. So it is present in all mammalian cells it is playing a major role in the cellular entry. So, after nanoparticles interact with the receptors on cytomembrane, a kind of cytosolic protein named clathrin will polymerase on the cytosolic side of the plasma where the cargo is internalized. So, after wrapping the nanoparticles inside, so the vesicle is pinched off through the GTPase activity of dynamine and forming a clathrin coated vesicles. So, with the energy supplied by the actin, this CCV move towards the inside the cells and the route is regulated by the cytoskeleton. So, this clathrin coat is shed off in the cytosol to release the therapeutic molecule. So, you can see here this is your nanoparticle, okay. So, it is coming and binding to the cell and this is a clathrin protein, it is attaching to this membrane and it is forming a clathrin coated pit, okay, and it is forming like this kind of vesicle, and once your nanoparticle is entrapped into this vesicle and the vesicle can be closed with the help of dynamine and this will be taken inside the cell and there your clathrin will be released and only the naked vesicle will go into the endosome and there it will be degraded and it will release the your therapeutic molecule to the cytoplasm or the nucleus. So, where is the destination of these vesicles? As I told you earlier, it may be associated with the receptor that nanoparticles ligands attach to. For example, if you have the low density lipoprotein particles and it will be internalized through the LDL receptor and it will be transferred to the lysosomes for degradation. But if you are having iron loaded transferrin, so that will be engulfed by transferrin receptor and recycled to the cell surface, okay. And this route can be blocked by inhibitors like chlorpromazine or a hypertonic medium or potassium depletion. So, by using these inhibitors we can inhibit the pathway CME pathway and we can understand whether our nanoparticle is following this CME pathway or not. So, next one is caveolate dependent endocytosis. So, this is a common cellular entry pathway. So, most of the pathogens including viruses, bacteria select this way to avoid the lysosomal degradation. So, as it can bypass the lysosomes. So, when you make the nanoparticles, if it is following this caveolate dependent endocytosis pathway, that is better because it is escaping from this lysosomal degradation. So, it can release the therapeutic molecule into the cytoplasm or the nucleus. So, here the caveolin is a protein exists in most of the cells and it play a major role in this caveolate dependent endocytosis. So, there are three forms of caveolin, caveolin 1, 2, 3, okay, and depends on the cell and depends on the application, this protein comes and play a major role. So, let us see the caveolate dependent endocytosis in details. So, once the nanoparticles or the virus which can come and interact with the receptors and that will induce the formation of flask shaped vesicles, okay. So, which are cut off from the membrane by dynamine and this caveolate vesicles traffic to fuse with the caviosomes or multivesicular bodies which have a neutral pH. So, these caviosomes containing nanomedicines move along with the microtubules to the endoplasmic reticulum. And here these nanomaterials in the endoplasmic reticulum penetrate into the cytosol, okay, and then enter the nucleus through the nucleopore complex. So, compared to the clathrin dependent endocytosis, this pathway takes longer time and has smaller vesicles in the process. And the nanomaterials taking this way is it will avoid the degradative fate and it will enhance their delivery to a target organelle such as endoplasmic reticulum or the nucleus. So, which is very important for improving the therapeutic delivery. For example, if you are making a nanoparticle for delivering anti-cancer drug or any other therapeutic molecule, so if it takes this caveolate dependent endocytosis pathway, so it can escape from the lysosomes and it can escape from the lysosome and it can reach the cytoplasm or the nucleus to increase the therapeutic efficiency. So, in this caveolate dependent endocytosis, so the nanoparticles will come and bind to the cell, okay, and mainly this cholesterol enriched membrane will be there at the receptor site, and here you can see here this is your this V shape, that is your caveolin protein, and another protein is cavin, it is a light blue color, okay, and it forms like a flask shaped caveolae, and this dynamine will come and help in enclosing this vesicle, and this vesicle will go and bind to this endosome and the material inside these nanoparticles will be released into the cytoplasm as well as the nucleus. So, the next process is macropinocytosis. So, it is commonly defined as a transient and this is a clathrin and caveolin independent growth factor induced or the actin driven endocytosis, okay. So, that can internalize the surrounding fluid into large vacuoles and it can be found in almost all the cells, okay, and with a few exceptions like brain microvessels, endothelial cells, okay. And this pathway is generally started with an external stimulation which activate the receptor tyrosine kinases. So let us see the macropinocytes in details. So once the virus or your nanoparticles come and attach to the cell, so it will activate the intracellular signaling, which leads to the production of actin filaments. This actin filaments will make like this protrusion, and this protrusion will cover your nanoparticles, and it forms a macropinosomes, and this macropinosomes will reach your endosome, and there it will be releasing your nanoparticles with a therapeutic molecule, and that can be taken up by the cytoplasm or the nucleus. So in this clathrin and caveolin independent endocytosis, so which is mainly relies on the cholesterol and requires a specific lipid composition. So according to GTPases, which play a major role in regulating this cellular entry pathway, so this clathrin and caveolin independent endocytosis is classified into ARF6 dependent, CDC42 dependent or ROA dependent. And here dynamin also play important role, but the mechanism is not completely understood. So let us see some of the examples. So the nanoparticles may enter the cells through different endocytosis and passive mechanisms. So depending on the physical properties of nanoparticles and the composition of the bilayer, okay. So the nanoparticles have the ability to utilize all 4 of the endocytosis mechanisms to enter the cells. So some of the examples we see here. So polystyrene latex nanoparticles smaller than 200 nanoparticles could enter the cells through CME mechanism, and the larger nanoparticles will enter the cells through the caveolin endocytosis. And the gold nanoparticle with the diameter of 3.4 nanometer can enter the macrophage cells through the phagocytosis, and 200 nanometer polymeric nanoparticles would enter the cells through caveolin endocytosis, and by changing the nanoparticle surface groups, and it can also enter the cancer cells by CME as well as phagocytosis process. So based on the surface charges, again the mechanism of cellular uptake will be varied. So here the nanoparticles do not necessarily undertake the endocytosis process to enter the cell. For example, silicon nanoparticles between 15 to 200 nanometer have been reported to translocate through a membrane through the passive mechanism. So if you want to study whether your nanoparticle is following endocytic pathways, so we can use the markers to study the intracellular fate of these nanoparticles. Some of the classical probers or markers known to be internalized through the specific endocytic pathway. For example, this LDL, this low density lipoprotein, and transferrin enter the cell through the clathrin dependent endocytosis. So they are commonly used as the markers for CMV. And also we can use this like cholera toxin or Siga toxin, okay, and even caveolin 1, so which are usually used as a markers for caveolin dependent endocytosis. And again we can also use the dextran is the marker for macrophagocytosis. So when you attach this dextran or this particular protein, and we can understand whether it is entering the cells through this particular pathway or not. And there are several dyes available for specific organelles in the cell. For example, lysotracker and lysosensor for lysosome, okay. So this lysotracker dye will stain only the lysosome, and they can be used to detect the co-localization of lysosome and the labeled nanoparticles with the confocal microscopy. So let us see in this picture, we have used this cancer cell lines, and it is stained with the lysotracker green, okay. So this lysotracker green will stain only the lysosome of the cells, and this is a epirubicin. Epirubicin is an anti-cancer drug, and which has the intrinsic red color fluorescence. And when we overlay these images, we can see here this anti-cancer drug is entering the nucleus, and your lysosomes, that is the lysotracker green, that is on the cytoplasm, okay. And here all the red fluorescent signals were mostly distributed in the nuclear region and did not merge with the green fluorescent signals of the lysotracker. So it shows that this nanoparticle is escaping from the lysosome, and it is entering into the nucleus to deliver the particular therapeutic molecule. So there are several inhibitors also available which can be used to block the specific endocytic pathway to confirm whether it is employed by the nanoparticles to enter the cells. So these are some of the examples like hypertonic sucrose or chlorpromazine and potassium depletion, so which can be used to inhibit the clathrin dependent endocytosis, and these chemicals can be used to inhibit the caveolin dependent endocytosis, and these molecules can be used to block the macrophagocytosis. So by using these kind of chemicals, we can block the particular endocytic pathway, and we can add our nanoparticle, and we can check whether our nanoparticle is following this particular pathway or not. And here the nanoparticles properties play a major role in this cellular uptake. For example, the nanoparticle size, shape, surface charge and surface functional groups, and also nanoparticles hydrophilicity. So these are the various properties which is going to play a major role in this cellular uptake. So let us see how the size plays a major role in cellular uptake. So the size of the vesicles that contain nanoparticles varies with the specific pathway, and the particle size should be small enough to enter the vesicles, and the size range from 10 nanometer to 5 nanometer and limited up to 5 micrometer. So depends on the size, the particle will take the different kind of cellular entry, and the larger particles are most likely to be engulfed by macropinocytosis, and the size of the vesicle involved in the clathrin mediated endocytosis is approximately 100 nanometer, and the size involved in caveolae mediated endocytosis is 60 to 80 nanometer. And let us see the shape. So until date there is no specific conclusion on the pathway selection of nanoparticles based on the shape. Some of the reports showing that rod shape particles have more cellular entry when compared to the spherical shape, but still a lot of research is going on, there is no proper conclusion. So at similar lengths, spherocylindrical nanoparticles have shown a higher lipid bilayer translocation efficiency when compared with the spherical or pyramidal or conical nanoparticles. And depending on the diameter of carbon nanotubes, the cellular entry of the carbon nanotubes can occur either passively or endocytosis. Okay. So with the smaller diameters reported to pass through the membranes passively, and again when you use the carbon nanotubes, okay, or nanorods, depends on the size and depends on the diameter of the nanoparticles or nanorods, so it can select the particular pathway to enter into the cell. So let us see the effect of particle shape on the phagocytosis. So this omega is an angle between the membrane normal at the point of attachment. Suppose if the omega angle is less than 45, so the particles can be internalized successfully, and if the omega angle is more than 45, the internalization of the particles can be inhibited. And let us see the role of surface charge. So here the positively charged nanoparticles can escape from endosomes after internalization, and it will exhibit a perinuclear localization because of the proton sponge effect. And here the nanoparticles without any charge at physiological pH may interact with the cells with the aid of hydrophobic and hydrogen bond interactions. So let us see what is the proton sponge effect. So the positively charged nanoparticles will attach to the cells and it forms a endosome, and these endosomes will fuse with your lysosome, okay, and it forms a endolysosome, and this endolysosome, due to the more amount of positive charge in the endolysosome, it will break and it will release the nanoparticle into the cytoplasm, and this nanoparticle can release the drug to the nucleus and it can induce the therapeutic effect. So this is called as proton sponge effect. And here the neutral particles coated with hydrophilic polymers can prevent the interaction with the cytomembrane, leading to less absorption, and anionic particles may be endocytosis through the interaction with the positive site of the proteins in the membrane, okay, and they can be highly captured by the cells because of their repulsive interaction with the negatively charged cell surface. So the positively charged nanoparticles can easily attach to the cells, as you know that cells have the negative charge, and these anionic nanoparticles, so that can be taken up by the positively charged protein receptor on the cell surface. So these cationic nanoparticles strongly interact with the membrane and enter the cells rapidly, and these anionic particles bind the positive site at the membrane and enter the cells, and again neutral nanoparticles can also enter into the cells through the various process. So here these cationic nanoparticles mainly enter the cells through the CME, and some research shows that they can utilize macropinocytosis or caveolae and clathrin independent endocytosis, or it can even follow multiple pathways including caveolae mediated endocytosis, and mainly the anionic nanoparticles, so these are likely to taken up by the caveolae dependent endocytosis, and again the neutral nanoparticles, there is no clear preference for specific routes. So let us see the role of surface hydrophobicity and hydrophilicity. So these hydrophobic nanoparticles have higher affinity for the cell membrane than hydrophilic ones, so leading to an improvement of cell uptake in the kinetics and the amount. And these hydrophilic polymers used to modified nanoparticles such as polyethylene glycol PEG or the PVP and dextran, so which form a cloud to suppress the interaction between the nanoparticles and the lipid bilayer of the cells. So thus it will increase the prolonged circulation of the particular nanoparticle in the system. And again, the chemical composition at the nanoparticle surface also determines the surface hydrophobicity and which influence the route of cellular uptake. And nanoparticles with different hydrophobicity present different affinity with the cell membrane. So you can see here, if you have the hydrophobic particle or the hydrophilic particle, so it will be taken up by the cells, depends on the affinity with the cell membrane. So as a summary of this lecture, in this lecture we have learnt what is the role of protein corona and what are the various multiple portals available for cellular entry of the nanoparticles, and we have also learned the details of phagocytosis and phagocytosis process, how the nanoparticles can use this process to enter the cell, and we have also learned how the nanoparticles properties influence their cellular uptake. So I will end my lecture here. I thank you all for listening to this lecture. I will see you all in another interesting lecture.